95% of the blood T cells have alpha beta T cell receptors. However, there are a small population of T cells which have gamma delta T cell receptors, and these cells are called gamma delta T cells. They contribute about 1 to 5% of the total peripheral blood T cells. As you can see, they have a gamma and a delta chain instead of a alpha and beta chain, and each chain has a variable and a constant region, a transmembrane region, and a cytoplasmic tail. The gamma and delta chain has an antigen binding site. However, the T cell receptors of the gamma delta T cells are limited in their diversity, and they are involved in recognition of antigens, which is MHC independent. So they are not bound to recognize antigens which are presented by MHC, and they are therefore not MHC restricted. So these gamma delta T cells play a role in response to different viral and bacterial pathogens. However, the ligands, specific ligands to the gamma delta T cell receptors are unknown. The cytoplasmic domain of the T cell receptors are very short, and they cannot transduce a signal to the nucleus. Therefore, they require the help of a complex called CD3 complex for transduction of signal to the nucleus. So these TCRs are binded to the CD3 complex by non-covalent interactions. A CD3 complex is formed by a number of different types of chains like gamma, delta, epsilon, and zeta. Together they form two heterodimer which are called CD3 and a homodimer which is made up of two zeta chains. The two heterodimer are the one which are formed by epsilon and delta, and the other is formed by gamma and epsilon. The homodimer contains both the same chain, that is zeta chain. These CD3 chains are invariant, that means they are constant or are identical in all T cells. So these CD3 complex are essential for expression of T cells during T cell development. So the signaling is initiated by aggregation of T cell receptors by MHC peptide complex on antigen presenting cells, which then activates CD3 complex, which results in transduction of signal to interior of the cell. Here you can see a structural comparison of B cell receptor and T cell receptor. B cell receptor is made up of 4 polypeptide chain, and T cell receptor is made up of 2 polypeptide chain. Both of them have a short cytoplasmic domain and are not able to transduce signal to the nucleus. In case of B cell receptors, they require the help of a heterodimer made up of Ig alpha and Ig beta, and in case of T cell receptors, they require the help of CD3 complex made up of 2 heterodimer and 1 homodimer. So when you look at the functional or differences or the differences in the properties, you can see the B cell receptors are able to recognize different types of antigenic molecules like lipids, proteins, carbohydrates, and the epitopes can be linear or conformational. However, the T cell receptors are restricted to recognition of only normally the peptides, which are presented by antigen presenting cells, and they recognize only linear short sequence of amino acids. So BCRs and both TCRs have unique specificity, that is they have the ability to bind to specific type of antigens, and when we make a comparison of both, T cells have more diversity as compared to B cell having 10 to the power 11 uh, clones. So signaling in BCR requires Ig alpha and Ig beta heterodimer, and in case of T cell receptors requires CD3 complex made up of CD3 and zeta subunits. B cell receptors also have an effector function that is formed by the Fc region of the antibodies. However, T cell receptors do not have any effector function.
T cell or T lymphocytes. T represents the term thymus, and we know that T cells are thymus-derived. T cell precursors originate in the bone marrow and they migrate to the thymus, to complete their maturation and become T lymphocytes. These mature lymphocytes then migrate to bloodstream and lymphoid tissue where they circulate. We also studied that when these T cells encounter antigens, they recognize those antigens by T cell receptors, which further activate the adaptive immune response. In this video lecture, we will study about T cell receptors and CD3, which is a signaling protein associated with T cell receptor. T cell receptors, abbreviated as TCRs, are molecules on the surface of T cells. These are membrane-spanning proteins. It is this TCR which interacts with antigens presented by MHC molecules on the surface of antigen-presenting cells or target cells. Each T cell has thousands of copies of the same TCR on its surface. An individual T cell generally carries between 10,000 and 30,000 identical copies of a single TCR. There are two types of cell receptors and both are heterodimers. First one is TCR alpha beta and second one is TCR gamma delta. 95% of the T cells circulating in the body have TCR alpha beta and 5% have TCR gamma delta. T cell either express alpha beta receptor or gamma delta receptor, but never both. Let's have a look on the structure of TCR alpha beta. TCR alpha beta is composed of two polypeptides, an alpha chain which has a molecular weight of 49 kilodaltons and a beta chain which has a molecular weight of 43 kilodaltons. Each chain has two extracellular domains, a transmembrane portion and a cytoplasmic tail. The outermost domain is known as variable domain and represented by capital V. Other domain is known as constant domain and it is represented by capital C. Each of the domain is stabilized by an internal disulfide bond. The variable domains of both alpha and beta chain together form the antigen binding site. After T cell receptors recognize the antigen presented by an MHC molecule, a signal is transmitted from the surface of T cell to the nucleus, which leads to T cell activation. But T cell receptor cannot do this alone because the cytoplasmic tail of the T cell receptor is extremely short. It is about 3 amino acid long. So, how this signaling takes place? For this, T cell receptors are associated non-covalently with a complex of proteins. This complex is made up of 4 different proteins, namely gamma, delta, epsilon, and zeta. 2 heterodimers formed by these proteins are known as CD3. First heterodimer is formed by delta and epsilon protein. And second one is formed by gamma and epsilon protein. Besides CD3, 2 zeta chains, linked together by a disulfide bond, forms the remaining part of this protein complex. So, here you can see the whole TCR complex on the cell surface of a T cell. The CD3 proteins and the zeta chain are identical in all T cells. It is important to note that the whole complex of proteins, that is, alpha beta of T cell receptor and gamma, delta, epsilon, and zeta, is transported to the cell surface as a unit. Experiments have shown that if any one of these proteins is not made, TCR is not expressed on the cell surface. So, in this video lecture we saw that T-cell receptor is actually a complex of proteins. The alpha and beta proteins of this complex do the job of recognition of the antigen. They cannot signal. For signaling job, gamma, delta epsilon, and zeta proteins are there. They initiate intracellular signaling pathway after antigen recognition by T-cell receptors.
It's time for our first concept checkpoint. The first question you have to find out what's the difference between MHC1 molecule and a T cell receptors. Both the differences in the structure as well as in the function. And second question is you have to find out, can T cell receptor recognize known proteinaceous antigens? Yes, no, how? You have to explain that. So try to find out the answer and we will discuss them in our Q&A session.